Hello my dear friends, welcome back to my YouTube channel. Myself I am Lokush Jogi. I have completed MSc in Agriculture, Seed Science and Technology, Specialization Seed Science and Technology from HNB Central University Srinagar, Uttarakhand in 2020. I have got 52 rank in PhD program Seed Science and Technology from Indian Council of Agricultural Research ICR All India Entrance Examination for Admission in 2021. Today I am going to tell about the topic seed production of fruit vegetables. Brinjal. Land requirements. There are no requirements as to previous crop, but the land should be free of volunteer plants. The soil of selected fields should be fertile, rich in organic matter, sandy loam and well drained. Isolation requirements. Brinjal is a partially self and cross-pollinated, but self-pollination is more common. The extent of natural crossing depends upon insect activity and has been recorded from 0 to 48% for pure seed production. Seed fields must be isolated from other variety brinjal fields and fields of same variety not conforming to varietal purity requirements of seed certification at least by 200 meters for foundation seed production and 100 meters for certified seed production. Brief cultural practices. 1. Time of sowing. Plants February to March, June to July, October to November, hills March to April. Sowing of the seed crop should be so adjusted that maturity does not coincide with heavy rains. The winter crops needs special protection from frost. 2. Preparation of land. Prepare the land to a good tilth by ploughing and 3 to 4 harrowings followed by leveling. 3. Source of seed. Obtain nucleus or waders or foundation seed from a source approved by the certification agency. For seed rate 375 to 500 gram per hectare. 5. Sowing of seed in nursery. Seeds are sown on raised nursery beds 15 to 20 centimeter high from the ground in rows 2 to 3 centimeter apart. 25 beds of 2 to 2.5 meter long and 1 to 1.5 meter wide will rise seedlings to plant 1 hectare. 4 to 500 handfuls of ammonium sulfate or CAN dissolved in 7 to 8 gallons of water can be sprinkled on nursery beds after about a fortnight of germination in order to get healthy and vigorous seedlings. Wash out the fertilizer immediately from the plants by sprinkling simple water. Thin sowing should be done to avoid dumping of the infected areas. Drenching with Captan at 150 gram in 100 liter of water to cover 200 square meters should be used. 6. Fertilization. Applied to 20 to 25 ton of well rotted farmyard manure at the time of the land preparation. 250 kg ammonium sulfate or CAN, 300 kg superphosphate and 120 kg potassium sulfate before transplanting time. For top dressing, use 250 kg ammonium sulfate or CAN in 2 doses. The first 15 days after transplanting and the second at flowering time spread around the plants. Irrigate immediately after top dressing. 7. Transplanting. Transplant the seedlings when 12 to 15 centimeter tall, preferably at evening time. Irrigated immediately afterwards at spacing A. Known spreading types 60 multiply 60 centimeter between row 2 and plants. B. Spreading types 75 to 90 multiply 60 to 70 centimeter. 9. Irrigation. Irrigate at 2 weeks intervals during the summer. 10. Interculture. Keep the fields free from weeds and the soil well irrigated by frequent hoeings. 11. Insect and disease control. Insect, fruit borer, shoot borer, Ephilacha, beetles, aphids, jacids. Spray 625 ml parathion in 625 to 750 liter of water. Repeat at 15 days interval with Nuvecron at 625 milliliter per hectare. Mealybugs, red spider mite. Spray 625 ml of parathion hectare. Spray Calathen at 250 ml per hectare. Disease, seed-borne disease. Treat seed with Thyram or Captan at 3 gram per kg seeds. Dumping of. Spray Captan at 1.25 kg in 800 liter to 1000 liter of water. Repeat if necessary. Little leaf. Control to the insect vectors by spraying either 625 ml of parathion or 625 ml of metacystox per hectare. Powdery mildew of leaf spot anthracnose. Spray Benlet at 150 gram in 250 liter of water per hectare. Osmosis blight fruit rot. Starting from immature fruit stage, spray 2 to 3 times Captan or Minev or 0.2% at 10 days intervals. Rogging. An yearly rogging for off types plants is possible since off types may be often be detected much before blossoming has occurred. For growers familiar with varieties to rogue out more of types plants later when each plant has several more or less mature fruits. Rogging can be based on fruit size, shape and color as well as the plant's overall performance. In addition to off types plants affected by disease such as osmosis, blight and little leaf etc. should be removed from the field from time to time. Harvesting and seed extraction. Harvesting is done when fruits are fully ripe. The fruit are picked and collected. The outer covering is peeled off and the flesh with the seeds is cut into thin slices. These are then softened by soaking till the seeds are separated from the pulp. If the material is allowed in this condition to stand overnight, the separation of seeds from the pulp become easier. After separation, the seeds are dipped into water. Those which float should be rejected. The seeds should be then be dried in partial shade to a moisture content of 8% or below before storing. Seed yield. The average seed yields is 100 to 200 kg per hectare. Hybrid brinjal seed production. In brinjal, hybrid seed production can be produced by hand emasculations and pollination technique as its floral morphology favors rapid emasculation and pollination. Also, a large number of seeds are formed in a single fruit. At 800 to 1000 seeds is long brinjal and 1000 to 1500 in round brinjal. Therefore, a very limited number of fruits produced in a sizable amount of seeds. Technique of hybrid seed production. In producing the hybrid seed, the variety setting, the large number of seeds in single fruit should be taken as the female percent so that a large amount of seeds could be obtained in a single attempt. The flower buds which are expected to open the next They are selected one female parents with the help of forceps. The flower buds are opened and the stems, the number of the which varies from 5 to 7, are removed one by one. This process of removing the stamens is called emasculation. The emasculated buds are then baked in butter paper or muslin cloth bags to prevent pollinations with the undesirable pollens. While emasculating to the flower, or should be exercised that no another is ruptured or crushed. If it happens, such flower should be rejected and the forceps should be sliced with spirit or alcohol. The flower buds of the male parents should also be baked to avoid contaminations. Next day, in the morning, the flowers which were baked for taking the pollen grains are plucked and collected in a petri dish. The female buds are then uncovered. The anther from the male flower is removed and is held in a bee between the arms of the forepaws. As the pollen grains in the anthers of Brinjal are released through apical pores, the anther is held perpendicular to the stigma surface, keeping the apical pores of the anther opposite to the stigma surface. The forepaws are tapped and the yellow colored powder of pollen mass is dusted on the stigma. The process of dusting the pollen grains on the stigma is known as a pollination. The pollinated buds are again baked to prevent cross pollination. The emasculation and pollination can be done simultaneously. However, the success in fruit sitting when this method of is flowered is marginally reduced, but the labor and time required for bagging to the emasculated buds are unbagging them the next day for pollination is effectively saved. The emasculated and unpollinated buds and male buds are tagged by tags of different colors so that each set of buds can be distinguished with the help of the color of the tags. Thanks.